Hello, dear students. This is revision short 5, linkage and recombination. This is a very important topic. This linkage and recombination was given by Morgan. Uh, the Mendel proposed the law of independent assortment, but later experiments showed that this law is not always followed. Thomas Hunt Morgan, while working on Drosophila melanogaster, we saw in the previous lecture, he discovered an exception which is known as linkage. Now, this Morgan, he performed dihybrid crosses involving sex-linked genes. So this is very important. Uh, he performed dihybrid crosses involving sex-linked genes. And we are talking about Morgan's work, which are located on X chromosome. Sex-linked genes on X chromosome. When he crossed yellow-bodied, white-eyed female flies, so you have here yellow-bodied, white-eyed female these flies with brown-eyed and what do we call it with brown-bodied and red-eyed male flies, he noticed that F2 phenotypic ratio did not follow the Mendelian ratio, that is 9 is to 9 is to 3 is to 3 is to 1. So it deviated. So this was his observation. So when Mendel crossed yellow-bodied, white-eyed female flies with brown-bodied, red-eyed male flies, he noticed this thing, the deviation in Mendelian ratio. Instead, parental combinations appeared much more frequently than new combinations. You can see here 98.7% parental combinations than the new recombinations. This indicated that the genes were physically located on the same chromosome and hence were inherited together. So Morgan called this phenomenon as linkage. He called it as a linkage. However, Morgan also observed that sometimes new gene combinations appear. This was due to crossing over during meiosis. And he called this as a recombination. Now, Morgan found two important patterns. He said that tightly linked genes show very low recombination. Tightly linked genes. Very low recombination, as you can see here, 1.3%. Example is white and yellow genes, which we have talked about. The recombination is 1.3% only. Now, loosely linked genes, they show higher recombination. Example is this one here, 37.2%. These are loosely linked genes. Now, Morgan had a student. His name was Alfred Sturtevant. This guy. He used recombination frequencies to measure the distance between genes on a chromosome and created the first genetic map. That's called gene mapping. Now you should know that how this gene map is formed, it is made. So it was Morgan's student, Alfred Sturtevant, who did what? Who used recombination frequencies to measure the distance between genes on a chromosome and created the first genetic map. Just remember this thing. Thus, what we conclude from here, the closer two genes are, the lower the recombination frequency, and farther apart they are, the higher the recombination frequency. This is very important. Now, when we look at this diagram, we look at cross A here. Cross A, tightly linked genes Y and W. Genes, that is the yellow body and white eye. Both genes are very close to each other on the chromosome, X chromosome. Okay. Result is 98% parental types and only 1.3% recombinants. Here the crossing over is rare, that's why this thing happens. So conclusion we can draw here is closer genes means stronger linkage, means less recombination. When we talk about cross B, what has been shown here is loosely linked genes, that's W and M. W represents white eye and miniature wing. And genes are far apart on the same chromosome. You can see here. It results in parental types, right? 62.8% parental types and more recombinants, 37.2 recombinants as compared to this one. So crossing over is frequent here because these genes are away from each other, far away from each other. So we can conclude from here, father genes means weaker linkage. It means more recombination. So this is a very important topic. So you should watch this video 2-3 times so that you can understand it properly. Thank you for watching.